Masters from Pune University. He has cleared all national level examination including CSIR, UGC, NET, DBT, JRF, ICMR, JRF, GATE, etc. all the national level examination. Sir has 14 years of teaching experiences including UG and PG students, and his research interest is structural biology and neurodegenerative diseases. He worked on model system C. elegans. He has successfully completed many projects from UGC, DST, and SCRB. Sir has published more than 20 research articles in various national and international journals. So, sir, on the behalf of Parul University, I would like to welcome you. Sir will be delivering his lecture today's webinar on gene regulation in prokaryotic organisms. So, gene regulation in prokaryotic organisms, this title is very important nowadays as students will be able to crack their national and international examination using this particular topic. So, sir, I would like to request you to please continue.
Yeah, thank you, Dr. Dhoni. So, good afternoon to all of you. Just a minute, I'm sharing my screen. So, Dhoni, can you see my screen?
Yes, sir. It is very well visible, sir.
It is visible. Okay, just a moment. Good afternoon to all the audience. So today's lecture is mostly concentrated about the the students' point of view. As Dr. Dhoni told me, the students from the BSc as well as the MSc, PhD and other students are also there, as well as the faculty members are there. So I've made my video, presentation more suitable to the students. And I request all the students, whenever or in between you are not getting the things, please ask me, right? So that we can make the things clear. Okay. The protein concentration, or we can say the homeostasis of the protein inside the cell, is always maintained. And this process or this maintenance of the homeostasis, just a moment, just now setting something. So this homeostasis or the steady state of the protein concentration is maintained by the different basic processes like transcription we heard about, right? Translation, and in many eukaryotic organisms, the post-translational, post-transcriptional modifications are there. And finally, we got the protein concentration or the homeostasis of the protein inside the cell. So the important thing about these protein homeostasis is that how it is regulated. Many proteins are not always needed. Some are needed at a certain time interval. Some are not needed at that time of interval. So always there is a process of switching on and off, right, of the genes inside the prokaryotic cell. Here we say, because our lecture is concentrated about the prokaryotic system only. So the gene expression is mostly regulated at different fundamental processes we have seen in the previous slide, and mostly the metabolic conditions, the physiological conditions, environmental conditions, and etc. Many more other conditions are also there, but these three conditions are the major condition where the gene expression has been regulated. And in prokaryotes, mostly the gene regulation is regulated at the transcriptional as well as the translational level. But if we talk about the other organisms like eukaryotic system also. So the processing as well as the transport of mRNA from the nucleus to the cytoplasm, at that level also it is regulated, right? So now we concentrate about the prokaryotic system. So in the prokaryotic system, the gene regulation or the control of gene regulation, we can say it is mostly at the transcription initiation level, right? Mostly, not always, okay? So transcription initiation level is the very favorite site to regulate the gene expression in the prokaryotes. So first of all, we discuss basic things about the gene and how it is transcribed, how the RNA polymerase binds over there. Suppose this is the structure of a gene, right? So these three are the structural genes, let's say A, B, and C. Okay, and this is the regulatory element, or we can say the promoter. And here the RNA polymerase binds, that we know very well. Okay, so RNA polymerase binds here, and that RNA polymerase will then move on to the DNA, and consequently it synthesizes the mRNA of A, B, as well as the C, or the genes located downstream to the promoter, right? Okay, so the RNA polymerase here, we can say the RNA polymerase here is responsible for the RNA polymerase is nothing but a protein, right? So this protein is, making the things capable to synthesize the RNA, right? So we can say whenever the RNA polymerase is present, so the gene, or we can say the transcription takes place, right? But the many other proteins are also there inside the cell, which inhibits the synthesis of the mRNA, or after the mRNA, I mean, at the level of translation also. So we categorize these regulatory elements broadly into the two different types, right? One is positive regulator and another is negative regulator, right? So first of all, we try to understand these two terms very clearly. So positive regulators are nothing but biologically we can say, or scientifically it can be said as activators, and negative regulators it can be said as repressor. All right. In the next figure, we try to understand the positive as well as the negative regulator, right? So this panel, the left panel is the negative regulator, and the right panel is the positive regulation. Okay. So yeah, you can ask me, Nikki. You are raising your hand, so you can ask me a question. Yeah, if you have any question. Okay. So negative and the positive regulation. So see, the very simply, this is the structural gene, suppose, and this light blue color is the promoter region. This region is promoter. Okay, and if this pink protein, is bound here, right? So at that time gene expression is not there. That means RNA synthesis is not at all. Okay, but as soon as this pink protein will leave here, it leaves the promoter site. Okay, transcription takes place immediately. See, if this protein is left the promoter, then transcription takes place. What we make the conclusion here. So as long as this protein is present, no transcription is there. But as soon as this protein is left the promoter, transcription takes place. That means in the presence of this pink protein, the transcription won't be there, right? That means this protein is inhibiting the process of transcription, right? So it can be called as negative regulatory element, right? Negative regulatory element we can say. All right. Another example here in the B figure. Again, this protein is present along with the signal molecule here without signal molecule, right? And here with signal molecule, some other molecule you can consider, right? And as soon as this pink protein leaves the promoter, the mRNA synthesis takes place, right? So this panel represents the negative regulation, right? Another or right panel here we can see when this green protein is bound, right, somewhere near to the promoter region, right? This green protein facilitates the binding of the RNA polymerase. See, it promotes the binding of the RNA polymerase, and the process of transcription takes place very smoothly here. So what conclusion we can made here? This green protein facilitates the transcription, isn't it? So the entire process can be called as positively regulated process, because this, in the presence of this protein, the mRNA synthesis takes place. All right. Here also you can see this green protein is present, right? Along with the signal molecule. Sorry, the meeting is being recorded by continuing the webinar. Yeah. Okay. So here also the green protein is there, right? And this green protein, it facilitates the binding of the RNA polymerase, and consequently the transcription takes place. Okay. So presence of this protein, it facilitates the binding of the RNA polymerase, and consequently the process of transcription takes place. So this can be considered a positive regulatory element. Okay. So negative regulatory element and positive regulatory element. I think you understood the things well, because we'll use this concept in the later slides. All right. So now we move on to the lac operon, the model which was discovered by the Jacob and Monod in early 1960s, right? They awarded the Nobel Prize also. And first time Jacob and Monod, they found out the gene regulation is at the transcription level, right? Transcriptionally genes are regulated. First time they have seen, right? And finally they gave, or they come up with a model, and that model is called operon. I mean, lac operon model, right? Since they were, working on a bacteria which utilizes lactose, or they were working on the lactose metabolism. So they referred that operon as a lac operon, right? So this operon, it comes from a English word that is opera. You must heard about this opera. Opera is nothing but a program in which singing, dancing, orchestra, many more things are going on simultaneously, but by the control of a single director, right? A single director or single person would be there, or producer or director, whatever we can say, right? So single director, it directs the dancing, playing, music, right? Everything, right? So many more genes here also has been controlled by a single set of regulatory elements, right? Jacob and Monod first time they found out, right? Earlier the concept was that a single gene, suppose this is a gene, so this gene is controlled by a single regulatory element. Suppose this is a regulatory element, right? So this, suppose this is operon, operator, right? So this operator is regulated by a single, suppose X gene is there, right? So one regulatory element can regulate only one gene, right? But they found out, yeah, that is not the case, right? They found out, no, many more genes can be regulated. Here in the case of lac operon, they found 3 genes, right? 3 genes are regulated by a single set of regulatory elements, right? And then they gave the concept of polycistronic, very, very important term actually, right? So they gave here the concept of polycistronic. I mean, earlier it was called cistron, right? Polycistron means many more cistrons, right? Many more cistrons are regulated by a single set of regulatory element, right? So polycistronic is structural gene, right? Unit of gene expression is called cistron, right? So 3 terms are there: cistron, muton, racon, right? Danger they gave, right? So this is cistron. So now they come up with the concept of polycistronic structure, right? Okay. And after the discovery of the lactose operon, then many more operons has been discovered, like tryptophan operon, arabinose operon, histidine operon. Many more operon has been discovered. All right. Okay. So now we look at the structure of the lac operon. So here, what the Jacob and Monod discovered, 3 genes are there, 3 structural genes are there, as we discussed. So these structural genes were called Z, LAC Y, and LAC A, right? And these structural genes are under the control of a single operator. This is OLAC, right? And this is the promoter where the RNA polymerase binds, right? These three genes here, they synthesizes their RNA, mRNA, and finally they produce protein. So LAC-Z, it produces a protein which is called beta galactosidase. Then LAC-Y, it synthesizes galactoside permease, or simply we can say permease. And third, gene A, it produces a protein which is called galactoside transacetylase, right, or simply transacetylase. Okay, the function of the beta galactosidase is that this converts, or this breaks down the lactose into the glucose and galactose. We come to this point in the next slide, the function of the different genes. Okay, so this is the one thing. Another important thing is that this entire cystron, or this polycystron, this entire gene of the lactose metabolism, is again regulated by a separate operon that is called LAC-I, right? So that this LAC-I has their own promoter, and it synthesizes a protein which is called LAC repressor, right? And when LAC repressor tetramerize, right, the 4 units of the LAC repressor will come together, then it becomes active LAC repressor, right? And then they evolve with a binding motif at the operator region, right? When they tetramerize, they come up with a binding domain or motif, right? And that domain can interact with the operator site of the lactose operon. So this active like repressor have capacity to bind at the operator. Okay, keep in mind. So now we talk about these three enzymes. So the first enzyme permease here. Okay, first enzyme is beta galactosidase. Okay, no problem. So beta galactosidase, what it do actually? So it breaks the lactose inside the cell. Suppose lactose is there. So this lactose can be broken down into the glucose, galactose, and glucose, right? I change the color of this pen red. Okay, so this is the galactose and glucose, right? So lactose is broken down into the galactose and glucose by beta galactosidase. All right. Another important function of the beta galactosidase is that some molecule of the lactose is converted into allolactose, right? So two functions they are performing, right? First, the lactose is breakdown into the glucose, galactose and glucose. And another function is that they convert the lactose into its isoform. This is the isoform of lactose only. This is nothing but the isoform of lactose only into the allolactose. And this reaction is actually reversible. This reaction is reversible whenever the amount of lactose is less inside the cell. So allolactose again converted back into the lactose, right? You keep in this point in your mind. So this is the beta galactose. Another enzyme which is called permease. So permease is nothing but the enzyme which makes the cell membrane permeable, right? So that this disaccharide, lactose disaccharide, big molecule actually, they are not able to enter inside the cell without permease molecule. So permease makes the cell membrane porous, right? Or it facilitates the entry of lactose inside the cell. And third enzyme, which is called transacetylase, the function of transacetylase is not very clearly known actually till now, but the function of information about the transacetylase is that they detoxify the toxins during the lactose metabolism, right? But the exact function of the transacetylase has not been known till now. All right. Okay. So now the thing is that how this operon is switched off and on. Suppose I'm telling you, when the we are feeling hot, we are feeling in summer we are feeling hot, then we try to start our AC. Otherwise, we won't start RAC. At winter, we are not going to start RAC. Why? So the reason is that when the environmental conditions, when the environment is hot, right, then we start RAC, right? Similarly, inside the cell, when the when a particular protein is required only when they are switched on, otherwise it will become switched off. I mean, gene becomes switched off, right? So on off process is always going on inside the cell depending upon the requirement of the requirement of the protein, particular protein. All right. Some genes are always, in the condition of switched on actually. So those are called housekeeping genes, right? Housekeeping genes, right? So that is a big, that is another family of genes. So they are not switched off and switch on the regular basis, but most of the gene they switch on and off depending upon the requirement. All right. So first scenario, we try to understand when the lac operon is switched off. So this, as we discussed earlier in the back slides, that lac I, it synthesizes a protein which is lac repressor. So this lac repressor monomer, when tetramerized, then it becomes active, and when it becomes active, it will bind at the operator site. And when it binds at the operator site, in that case, RNA polymerase is not able to move, right, through the operator to the ZYA. They are not able to move, right? So there is no transcription takes place. And if no transcription is taking place there, then no translation would be there. So no proteins. I mean, all three proteins are not there. So the gene is considered as switched off. All right. Another condition, when is supposed to switch off. So see what we are discussing here. We are discussing here the lactose operon. I mean the genes involved in the lactose metabolism, right? So when you add lactose inside the medium, right? When you add lactose inside the flask, right? Inside the flask, bacteria is there, and you add lactose, right? Lactose in the medium. So bacteria is, and no other carbon source is there, only lactose is there. So bacteria will try to take the lactose only because no other carbon, no easily available carbon sources are there. So what it do actually, it try to take the lactose inside the cell, and then they get glucose and finally the ATP and whatever they want actually, right? So see, another scenario is that when the lactose would be there, then they have to break down the lactose into the glucose and galactose. So they need beta galactosidase, right? They need permease because lactose is the disaccharide. They are big molecule. They are not able to enter inside the cell, so they need permease also, right? So the gene is switched, should be in the switched on condition. All right. So for making the switch on, what happens here? So this lac I, it synthesizes repressor, they tetramerize, right? But in the meantime, here the lactose is present in the medium. So if lactose is present, we already have seen that lactose, they form their isomer, which is isoform, that is called allolactose, right? Allolactose. So these allolactose will bind with the tetramer when the lactose is present. When the lactose is present in the medium, right? And of course, we kept our bacteria into the lactose containing medium. So lactose, it converts into allolactose, and allolactose will bind to this tetramer. And when the allolactose will bind with this tetramer, they distort their domain or the motif, which is responsible for binding at the operator site, right? See, every binding and unbinding, right? These all are depending upon the conformation, right? Right? Conformation means it's three-dimensional structure. So after binding of the allolactose here on the tetramer, they distort the structure. I mean, they lose the binding motif or the domain of the tetrameric or active repressor, which can bind at the operator site. So now it is not able to bind at the operator site. So if it is not bound at the operator site, so the RNA polymerase binds here. See, this is the RNA polymerase, right? And RNA polymerase will move through the operator to the ZYA, and it synthesizes mRNA, ZYA, and finally into the proteins, respective protein, right? So here, in the presence of lactose or in the presence of substrate, right, the gene is, they make the gene switched on, right? So what we call here the lactose operon is inducible operon, right? So lactose is induces the operon to switch on, right? So maybe they ask in the exam CSIR net or JNU entrance examination that which of the following is inducible operon, right? So lac operon is the inducible operon because the lactose, it induces the operon to switch on. Okay. Okay. Another thing written here is the IPTG. This is very important actually. IPTG is nothing but a synthetic molecule which is very similar to the allolactose. Okay, it is very similar to the allolactose, but this is synthetic molecule, and this IPTG cannot be catabolized by the bacterial cell, whereas the allolactose can be catabolized, right? So if you set a experiment by taking the allolactose, so this allolactose finally, when the lactose concentration goes down, so allolactose converts back into the lactose, right? So they their repression or their binding with the tetrameric repressor is not, because they are not in the sufficient quantity. So in that case, whenever we are doing some experiment related with the lac operon, or when we express our protein, we induce the the recombinant gene by the IPTG, right? So IPTG is a isopropyl thiogalactosidase, right? It is a synthetic molecule and it is not utilized by the bacteria. That's why we use this inducer, right? So this is the inducer we use normally during the time of gene expression. Okay. So now, if you understood, till here, you can answer me this question, right? The question is that I'll question and I'll answer the question here. So question is how the very first molecule of the allolactose has been synthesized inside the cell, right? I mean, what we learned till now, the bacteria, inside the bacteria, lac operon is there. Suppose this is a lac operon, right? Lactose is outside. This L is the lactose, right? Whenever lactose is not enter inside the cell, they won't convert into the allolactose, right? Because lactose is outside. And we have seen that lactose can enter inside the cell only when the permease is there, and permease is there inside. Permease तो यहाँ पे है ना? Permease, if gene is switched on, even though the permease is inside, outside no permease is there, right? So how the lactose comes inside the cell? This is the question, right? I mean, how the first molecule of the allolactose has been synthesized within the cell? See, the answer is very simple actually. What happens? We already discussed the binding of the any protein with the DNA, or protein or protein DNA interaction is not the permanent interaction. This is the result of the Brownian movement actually, right? So random collision is there inside the cell, and during the random collision, what happens actually when the RNA polymerase, suppose this is the RNA polymerase, and this is the, suppose here the green one. So this is the operator, and here the tetrameric repressor is bound, right? The condition is that inside the cell. So what happens actually? These bind, I mean these RNA polymerase and this tetrameric repressor bound at their respective site, but this binding is not permanent binding, right? They are not bounded here for the lifetime. The binding and unbinding is a they are constant process. The because they are in the random collision, so sometimes they bound here, sometimes they come out from here. This also, this comes out, sometime they comes to bound over there, right? So the scenario comes in thousands or more than thousands. A scenario comes when the tetrameric repressor is not present at the operator site. They are not present. I mean, they left due to the Brownian movement, And in that meantime, the RNA polymerase crosses the operator site and it comes to transcribe the ZYA gene, right? So what happens actually? The basal level of all the protein, right? Basal level of transcription always be there, right? जो भी चीजें हैं बेसल लेवल हमेशा मेंटेन रहता है सेल के अंदर, राइट? So here the basal level of transcription is always there. Matlab all the three enzymes are there in the very basal concentration inside the cell, right? And that basal amount of permease here is now able to bind at the cell membrane, right? And then they makes the way open for the entry of the lactose molecule, right? So this kind of expression is called leaky expression, right? So by leaky expression, the basal level of all the three proteins are maintained, right? And the things will be running as it is, right? Okay. So if you understood the things, you can try to solve this question, right? See, the question is that just I'm reading and I'm solving the question, but you also try to solve the question simultaneously. E. coli mutant isolated from the genetic screen showed following classes of mutation. The first mutation is there at the Lac-I gene, right? Three statements they are giving, four statements in fact, A, B, C, D, four statements they are giving, and they are giving the correct answer choices. First is the point mutation at Lac-I gene, right? Lac-I, that means the regulatory gene, okay? So if the Lac-I mutation is there, then what happens? If the Lac-I mutation is there, then they are not able to form the monomer properly, or proper monomer has not been formed. Which kind of mutation is there? That is different issue, but they are not able to form the proper monomer, right? Okay. So if proper monomer is not there, then they won't be able to tetramerize. Tetramerize nahi hoga. And then if it is not tetramerized, then they are not able to bind at the operator site. Okay. Operator site पे bind नहीं होगा. This is one thing. Okay. Second, the deletion immediately downstream to the transcription start site, right? I mean, just at the operator site. What they try to say, the deletion has been made at the operator site. So operator site, if deletion is there, second case is that if deletion is there, then at the operator, in that case also this tetramer, if active tetramer has been there, then even then they are not able to bind at the operator site because the mutation is there at the operator site. Second point they are saying. Third point is that duplication of the Y, lac Y, and fourth is a duplication of the A gene, right? And then now question starts. Choose the option which is likely to result in the constitutive expression, right? Constitutive means continuously, okay? Constitutive expression would be there. So say in this case, constitutive expression is there, right? Because the monomer or tetramer is not forming properly, so they are not able to bind at the operator. So gene expression will carry on as it is, right? Second case is that when the deletion is at the operator site, again, operator site, if it is deleted, I mean, if it is mutated, then the tetramer is not able to bind at the operator site, right? So it is constitutive, and there is no effect of Y and A, right? I mean, duplication of Y and A on the constitutive expression of the lac operon. So the correct answer would be the A and B. Both A and B are correct situation. All right. I hope you are getting the things. Right. Someone are raising the hand. If you have question, you can ask me in between if your host is giving you the permission. Right. Okay. This is the second question. Quickly, just I'm telling you. Yeah, Jibin Sebastian, if you have a question, you can ask me. If host is allowing, then I don't have any problem.
She is not asking the question. She was saying the answer, and it was the correct answer, both A
Okay,
and B.
okay, right. Okay, good, great. Okay, okay. So
Jibin has a question, sir. He wants to know what is consecutive regulation once again. It will be explained to you after some time.
Yeah, yeah. Constitutive expression is nothing but the, I mean, constitutive expression is nothing but the continuous expression. Constitutive means, I mean, no regulation is there, right? Then it, like, I mean, housekeeping genes, suppose housekeeping genes, they are expressing 24/7, right? So they are called, or they can be called as constitutive gene. Okay. I mean, no control is there, constitutively expressed, always expressing, 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 right? So that is constitutive. Okay, so the second question is that the culture A and B is taken. Actually, two cultures of bacteria has been taken, and A was, earlier grown into IPTG containing medium, right? A was grown in the IPTG containing medium, and then both A and B were taken and they grown into the lactose containing medium, right? A was pre-exposed with the IPTG, but B was not pre-exposed with the IPTG, right? So the question is, the culture rate is so this region behind this is okay. So what they say, both the culture was now used to inoculate into the fresh medium containing the suboptimal concentration of gratuitous inducer, right? So IPTG is there in the second medium also, right? So it was observed that culture B was unable to utilize lactose. Okay, the culture B was not pre-exposed, and it was seen that in the second medium, the B was not able to utilize the lactose. So they are trying to ask what is the region actually. So region is nothing, just the A was, since A was pre-exposed, A was pre-exposed to IPTG. Okay, so what happens here? The ZYA, all the three genes are expressing, right? तीनों के तीनों जीन यहाँ पे एक्सप्रेस करेंगे और प्रोटीन बनाएंगे, right? All three proteins has been made, but in the B culture, it was not pre-exposed. So now in the second time culture, now the B, they try to start the synthesis of ZYA protein, right? That's why the B is not able to utilize lactose properly, or as compared to the A. A will utilize the lactose very properly as compared to B, just because of the pre-exposure, right? So this is the question, right? And certainly the answer would be the culture A, the lactose permease was induced to a high level during the pre-treatment of the IPTG, of course, which allow the preferential uptake of the lactose. Okay. So now just this is the trailing slide actually for your information here. So this LAGZ, what LAGZ we studied, the beta galactosidase. The beta galactosidase is a very important gene for making the selection actually, right? We make or we select the recombinant and non-recombinants by the blue-white selection mechanism, right? So in the blue-white selection, what we do actually, so the basis is the LAGZ gene, right? So see, if the LAGZ gene is intact, right? So if the LAGZ gene is intact, so it synthesizes beta galactosidase, right? And as we discussed earlier, this beta galactosidase, it breaks the lactose into glucose and galactose. But scientists, they made another compound that is called X-gal. X-gal is a chemical which is synthetic chemical, and this is very much similar to the lactose, right? And this X-gal has also been broken down into the two different compounds, right? And after the dimerization, it gives a blue precipitate, right? So see what happens, those colonies have been intact LAC-Z gene, right? Intact LAC-Z gene, they convert the X-gal into the blue color colonies, right? Here on the plate, you can see some blue color colonies are there and some green color colonies, some white colonies are there. So blue color colonies are the recombinant, we can say. Suppose your restriction site is here. Suppose your restriction site is here. you try to clone your gene of interest here, right? So if it is not insertionally inactivated, if your gene of interest is not cloned here, right? So it gives blue color, right? That means non-recombinant, it gives blue color. But when the recombination takes place, when your gene of interest is kept here, then they won't synthesize beta galactosidase, right? So no such conversion of X-gal into the blue color precipitate will be there, right? So this is the basis of the blue-white selection. Now we discuss bit detail of the lac operon here. So here the first we discuss about the lac promoter. So as we have seen, the lac promoter is there in between the the lac I gene and the operator gene. Okay, so here the lac promoter is considered as weak promoter actually. In the lactose operon, the lac promoter is a weak promoter, right? Why? So if you make a blast, right? If you compare the consensus sequence of the promoter, this is the consensus sequence TATAAT at -10, and this is TTGACA. This is at the -35 region, and this is the lac promoter sequence. If you compare, this is not exactly similar. Actually, bit differences are there, right? And this makes the lac promoter weak promoter, right? This is the strong promoter. जितना इसके करीब होगा सीक्वेंस उतना strong होगा promoter, right? But this is not exactly similar. Many more variations are there, so this is weak promoter. But there is a site upstream to the promoter region. This is called CRP site. See, this is the site which is called CRP site. This is the promoter region of the lactose operon, right? This is the operator. And upstream to the promoter is the CRP site. Here, a protein which is called CRP CMP complex. See how this complex comes, we discuss in the later slides. So when the CMP CRP complex will bind at this site, then it becomes a strong promoter, right? Otherwise, the lac promoter is weak promoter, but when this comes into the picture, right, it becomes a strong promoter. All right. So this CMP CRP complex makes the lac promoter strong promoter. All right. Okay. Now discuss a bit about the lac operator. So we have seen between the promoter and the structural gene operator is there. We have seen only one operator, right? But actually three operators are there actually. This is the O1, this is O1, this is the O2, and this is O3, right? This is the distance actually, how far it is from the O1, right? So this is, I mean, O2 is there inside the structural gene itself, see, plus 412, right? So this is inside the structural gene. And all the three operator, it works in a cooperative manner, right? Operator is the site where the tetramer or lac repressor will bind, okay? So, but the main promoter here is the O1. O1 is the main promoter, and these two promoters, O3 and O2, they cooperate the O1. So this is the comparison just, they compared, if the O1, O2, O3, all three are there, then the fold of repression would be 1300, right? If the O1 and O3 is there, then 440 is there. Then O1, O2 is there, then 700 is there. If O1 alone is there, then 18 is there. And see, if O1 is not there, here no O1, no O1, no O1. So the repression fold is very, very low. So O1 is the main promoter, right? Okay. And all three promoters are working in a cooperative manner, right? See, this is the three promoters, how they are binding, making a loop. By making a loop, they bind with the repressor protein or repressor tetramer. All right. Okay. So now we what we discussed earlier, some proteins are negatively regulated, or some genes are negatively regulated and some genes are positively regulated. Some proteins are negative regulator protein and some are positive regulator protein. So we try to apply that knowledge for the regulation of the lac operon here. So the lac operon, first we try to understand negative regulation, how the lac operon is negatively regulated, right? Negatively regulated. So we just take a scenario here. The scenario is that when the lactose is not there, right? We take two scenarios, when lactose is there and when lactose is not there. So first scenario we are taking first, right? So the first, the lactose is not there. If lactose is not there, we have seen earlier. So no allolactose would be there, of course. So if no allolactose is there, then repressor will bind to the operator. Here you can see in the figure, this is the repressor, this is the tetramer. No allolactose or IPTG is there. Allolactose यहाँ पे नहीं है, right? So it will directly bind at the operator site. ये operator site पे bind कर रहा है, right? So if it is bind at the operator, so no RNA polymerase can move, can cross, so no transcription is there, right? So this is switched off. We can say this is switched off. So now you see here very carefully, due to the presence of this tetramer, the entire operon is switched off. That means presence of this protein makes the operon switch off, right? So this is negative regulator. This protein is negative regulator. All right. Now another situation we try to understand. If the lactose, okay, not lactose, if the glucose is there, another situation, second scenario will take if the glucose is there. If glucose is there, so the glucose, what it do actually, if the glucose is there inside the cell, it makes the pathway which is responsible for the adenylate cyclase is ineffective. Adenylate cyclase, I mean adenylate cyclase is an enzyme which makes the CAMP, isn't it? CAMP banata hai, adenylate cyclase. So if glucose is there, so no adenylate cyclase is there. So you try to understand the correlation, right? Then only you can understand this slide as well as the next slide. If glucose is there, यदि ग्लूकोज है सेल के अंदर, तो क्या होगा? जो एडिनाइलेट साइक्लेज है ये नहीं बनेगा, है ना? It won't be synthesized inside the cell. Or if adenylate cyclase is not there, then no CAMP would be there. ये नहीं बनेगा. All right, correct. So the scenario is like that. If the, I mean, we are trying to understand the positive regulation. So if the glucose is absent inside the cell, then scenario is opposite. यहाँ के just उल्टा होएगा. So adenylate cyclase is activated. Okay, कब? When the glucose is not there, right? So when adenylate cyclase is activated, then CAMP is present, right? If CAMP is present, then it can make a complex with CRP, as we have seen CMP CRP complex. So CRP is always there inside the cell, right? When CMP is available, then CMP will bind with the CRP and it forms a complex, CMP CRP complex, right? And this complex, if this complex is formed, see this complex is formed, then what happens? It facilitates the binding of RNA polymerase, see here, and it makes the transcription very smooth, right? So what we can see, this green protein or this CMP CRP complex is the positive regulator. मतलब इसके present होने से सब कुछ सही तरीके से चल रहा है ना? जैसे तुम्हारे क्लास में जब टीचर आते हैं तो सब कुछ सही हो जाता है अपने आप, सब पढ़ाई करने लगता है ना? तो like that. So here what happens actually, when this protein is there, then the expression of all the three genes, it is, I mean the entire operon is switched off, on, sorry, okay? Switch on ho jata hai. So that is positive regulation. So this is positive regulation. So this is negative as well as the positive regulation, isn't it? Now we try to solve this question, right? Then we proceed ahead. So the question is that in the lac operon, E. coli is controlled by both lac repressor, tetrameric repressor that we studied, right, as well as the cap. I mean, this is catabolite activator protein, CMP-CRP complex. Cap means CMP-CRP complex is called cap actually, catabolite activator protein, right? So in an vivo experiment, which lac operon, the following observations are made, right? The first observation, CMP level are high. CMP level high कब होएगा? ग्लूकोज से इसका क्या कोरिलेशन है? जब ग्लूकोज क्या होगा, बहुत बहुत कम होगा, है ना, या ग्लूकोज नहीं होगा, है ना? If glucose is not there, then only CMP would be there. पहली चीज. Second observation, repressor is bound to the allolactose. I mean allolactose कहाँ से आएगा भाई? तो लैक्टोज यदि होगा सेल के अंदर, if lactose is there, then it converts into the allolactose Okay. Third, cap is interacting with the RNA polymerase, right? Cap is interacting with the RNA polymerase. No, cap is not interacting with the RNA polymerase. Yeah, they are interacting actually. Cap, cap and cap, I mean CMP CRP complex can interact with the RNA polymerase. Of course, yes. So question is that which one of the following conclusion is most appropriate based on the above observation? Okay. सो ग्लूकोज एंड लैक्टोज इज प्रेजेंट, नो बिल्कुल नहीं, क्यों? क्योंकि यहाँ पे हमने देखा ग्लूकोज वुड नॉट बी देयर, देन ओनली सीएमपी लेवल विल हाई. Glucose is present, again this choice is wrong. The third choice, both are absent. Okay, glucose would be absent but lactose would not be absent because lactose है तब तो yellow lactose बना ना, then only yellow lactose will form. Okay, so this is also wrong. So next choice is that glucose is absent. Yeah, this is correct. See here, glucose is absent, glucose नहीं होगा तभी CMP बढ़ेगा, and lactose is present. Yeah, so this choice is correct answer. Okay, I hope you are getting the things. Okay, so now the very important thing, whatever you learned till now, you can apply these concepts for different scenarios for the E. coli growth and maintenance, and you can correlate them with the operation of the lactose operon. So four situations, four scenario we'll see here. The first scenario, if the glucose is present, right? The first scenario is glucose is present and lactose is absent, right? ये सिनेरियो दे रहा है कि यदि ग्लूकोज होगा और लैक्टोज नहीं होगा तो व्हाट वुड बी द सिनेरियो. सी नाउ यू कैन थिंक वेरी केयरफुली that glucose when present, it inhibits adenylate cyclase, we have seen, so no CMP would be there, right? So what happens, the entire operon would be switched off, yes or no, है ना, स्विच ऑफ होएगा. दूसरी तरफ हम देखें लैक्टोज इज आल्सो एब्सेंट, सो लैक्टोज इज नॉट देयर, दैट मींस टेट्रामर, टेट्रामेरिक रिप्रेसर दे कैन बाइंड एट द ऑपरेटर, राइट? Tetrameric repressor they can bind at the operator, so the binding of the tetrameric repressor, it inhibits the operon, so this is negative regulation, isn't it? So this scenario is the, you know, the entire operon is switched off. और सिंपल सी सोचने वाली बात है कि यदि ग्लूकोज, ग्लूकोज इज अ वेरी सिंपल कार्बन सोर्स, बहुत बहुत सिंपल कार्बन सोर्स है, बैक्टीरिया को कोई परेशानी नहीं होती है, इजिली बिल्कुल आगे सामने थाली लगाया हुआ मिल गया पूरा का पूरा, तो खाना बनाने की कोई कष्ट वहां पे नहीं करेगा, है ना? हाँ, अब यदि आपको ग्लूकोज नहीं मिला, लैक्टोज मिला, तो बैक्टीरिया क्या करेगा, बैक्टीरिया विल ब्रेक डाउन दोज लैक्टोज इनटू द गैलेक्टोज एंड ग्लूकोज, राइट? बट यहाँ पे ग्लूकोज ही दे दिया है, बहुत बहुत इंटरेस्टिंग कार्बन सोर्स दे दिया है बैक्टीरिया का, सो एंड लैक्टोज है ही नहीं, सो एंटायर ओपेरॉन वुड बी स्विच्ड ऑफ. Second situation is glucose is absent and lactose is present, right? Glucose नहीं है ना, glucose is absent and lactose is present. All right. See the scenario. Both scenario we'll discuss. So lactose is present, so it converts into allolactose, then binds to the lac repressor. Allolactose बना तो lac repressor से bind किया, and if it is bind to the lac repressor, then lac repressor won't be able to bind at the operator, so it makes the switch on, right? So the operon is inducible operon. Okay, second situation, second बोला glucose नहीं है इसमें, if glucose is absent, right, then adenylate cyclase would be activated, so CMP would be formed, right? CMP CRP complex will form, so finally the positive control would be there, है ना? ये क्या हो गया, यदि ग्लूकोज है मीडियम में तो पॉजिटिव कंट्रोल हो जाएगा. ओके, ठीक है. Third situation is that this is very interesting and important situation for exam point of view and knowledge point of view also. See, this is very important. Glucose is present, but lactose is, sorry, just a minute. Okay, yeah. So glucose is present and lactose is present. See what happens. Glucose is present, मतलब यहाँ पे पूरी की पूरी प्लेट लगाई हुई है, थाली लगाई हुई है, right? लेकिन ऐसा समझो कि we are so hungry that we can have at least 5 chapatis, but उस थाली में 2 ही चपाती है, right? और उसके बाद हमको सारे चीजें दे दिया है बनाने के लिए किचन में, right? So what we do, first we'll have this plate, right, in which 2 chapatis are there, and then we cook, उसके बाद हम बनाना शुरू करेंगे। एग्जैक्टली यहाँ पे भी ऐसा ही होता है। व्हेन द ग्लूकोज इज देयर इन द मीडिया, सो फर्स्ट ऑफ ऑल बैक्टीरिया विल यूटिलाइज ग्लूकोज। क्यों यूटिलाइज करेगा, तो यहाँ पे देखो, inhibits the adenylate cyclase, ग्लूकोज रहेगा तो नो सीएमपी, नो सीएमपी सीआरपी कॉम्प्लेक्स विल बी देयर, देन आरएनए पॉलीमरेज बाइंड्स वीकली, है ना, तो ऑफ कोर्स वीक प्रमोटर होगा और बहुत कम वहां पे एंजाइम्स बनेंगे, सो इट इज ऑलमोस्ट स्विच ऑफ। ओके, बट जैसे ही ये खत्म होगा, हमने 2 रोटी खा ली, नाउ भूख तो लगी है अभी, सो व्हाट दे डू बैक्टीरिया, दे नाउ कम ऑन टू द लैक्टोज। अब वो लैक्टोज को प्रेफरेंस देगा, ये तो खत्म हो चुका है, प्रेफरेंस इसको दिया था, बट lactose now is there, so lactose converted into allolactose, right, they bind with the lac repressor and now the lac operon would be switched on, right। see यहाँ पे अब देखो 2 तरह का ग्रोथ कर्व मिलेगा। if you see, if you look at the growth curve of bacteria, you will see 2 different kind of growth curve is there। see, this is the one growth curve, just I'm changing the color so that you can see it very clearly। see, this is the first growth curve। ग्रोथ कर्व हमने देखा है सिग्मॉइड होता है, तो यहाँ पे देखो ऐसा करके है और ये जस्ट प्लेटू में अब जा रहा है, खत्म हुआ यहाँ पे ग्लूकोज, ग्लूकोज विल बी एग्जॉस्टेड हियर, राइट, बट अगेन द लैक्टोज इज देयर, राइट, सो अगेन इट विल मेक अ लॉग फेज, फिर से एक लॉग फेज बना लिया क्योंकि यहाँ पे फिर से दूसरा सबस्ट्रेट मिल गया लैक्टोज। सो दिस काइंड ऑफ ग्राफ इज कॉल्ड डाईऑक्सिक ग्राफ, है ना, इसको हम लोग क्या बोल देते हैं, डाईऑक्सिक, ओ एक्स आई सी, आई थिंक दिस, ओके, सो दिस इज कॉल्ड डाईऑक्सिक growth curve। okay, so the case is that glucose is present and lactose is also present, right। fourth case is very very interesting, सबसे ज्यादा इंटरेस्टिंग यही केस है। see, if the glucose is absent and lactose is absent, तो कायदे से हम यहाँ क्या बोलेंगे भाई, दोनों के दोनों एब्सेंट है तो ऑफ कोर्स स्विच ऑफ होगा, हम बोल देंगे, है ना, लैक्टोज भी नहीं है, ग्लूकोज भी नहीं है, कुछ है ही नहीं, तो बैक्टीरिया खाएगा क्या, तो काहे को स्विच ऑन करेगा वो, ऑपेरॉन को तो ऑफ होगा। बट कैसे, तो इसको जरा हम लॉजिकली देखें। आंसर इज वेरी सिंपल, बट लॉजिकली यहाँ पे देखें क्योंकि लॉजिक हमको कुछ और भी बोलेगा। सी हियर नो लैक्टोज इज देयर, सो नो एलोलैक्टोज वुड बी देयर, सो नो रिप्रेसर बाइंड टू द ऑपरेटर, राइट, सो स्विच ऑफ, ये क्या हो जाएगा, स्विच ऑफ। ओके, दूसरी चीज को हम यहाँ पे लेके चलें कि जब ग्लूकोज नहीं है, क्योंकि यहाँ पे दे रहा है ना ग्लूकोज इज ऑल्सो एब्सेंट, ग्लूकोज भी नहीं है, सो ग्लूकोज इज नॉट देयर देन हाई सीएमपी वुड बी देयर। सी, यहाँ पे अब अपोजिट चीज हमको मिल रहा है, सो एक्टिव सीएमपी सीआरपी कॉम्प्लेक्स वुड बी देयर, हेंस इट इज स्विच्ड ऑन। कैसा होना चाहिए, स्विच ऑन होना चाहिए था, बराबर। लैक्टोज नहीं है तो ये कह रहा है स्विच ऑफ, लेकिन ये क्या कह रहा है, स्विच ऑन। अब होगा क्या भाई, होगा तो कोई एक ही चीज, या तो यस या नो, है ना। तो अब देखो, अब हम इसको देखें, ज्यादा हम फाइनली यदि देखें तो व्हाट हैपेंस एक्चुअली, दिस टेट्रामर, जो टेट्रामर यहाँ पे बना, राइट, जो टेट्रामेरिक रिप्रेसर बना, दिस टेट्रामेरिक रिप्रेसर विल बाइंड एट द ऑपरेटर साइट। ये टेट्रामेरिक रिप्रेसर बना, ये ऑपरेटर साइट पे पहले से बाइंड है, कितना भी यहाँ पे सीएमपी सीआरपी कॉम्प्लेक्स बन के आ जाए, राइट, लेकिन ये ऑपरेटर खाली नहीं है, राइट। सो आरएनए पॉलीमरेज, यदि हम ये आरएनए पॉलीमरेज बना रहे हैं, इट वॉन्ट मूव, राइट, ये मूव ही नहीं करेगा, राइट। सो अल्टीमेटली द ओपेरॉन वुड बी स्विच्ड ऑफ, राइट। सो क्या होगा, स्विच ऑफ होगा, स्विच ऑन नहीं होगा, ठीक है। आई थिंक यू गॉट द पॉइंट, ओके। So these are the 4 different situations, okay। So if you understood these different situations and till this slide, right, now you can able to solve this question, solve question. This is a very nice question actually. See, I'm helping you. So here I'm changing the color first. So see, 3 different experimental conditions are given here, right? Okay, so in one, it was grown in the medium containing glucose as a carbon source. First एक medium है, 3 medium है जिसमें पहला glucose है. Second is the medium containing both glucose and galactose, right? Glucose and galactose. And the third was infected with the phage virus से infect करा दिया bacteria को. Match the curve shown below to the treatment. Okay, see, बहुत simple. You can solve this very easily. This is the diauxic graph. ये जो हमको दिख रहा है ना, ये diauxic, ये नेट में पूछा हुआ क्वेश्चन है, नेट की एग्जाम होती है ना, ये देखो डायोक्सिक है, दिस इज द डायोक्सिक. सो ए वुड बी द ग्लूकोज एंड गैलेक्टोज. ए क्या होगा? ग्लूकोज एंड गैलेक्टोज, राइट. ओके. द ग्लूकोज में यदि ग्रो कर रहा है ना, सेकंड इज ग्लूकोज. सो सी, ग्लूकोज में द लैग फेज विल कम, राइट, एंड फाइनली एक सर्टेन टाइम के बाद इट विल एंटर इनटू द प्लेटो, राइट. एक प्लेटो, आई मीन ग्लूकोज है जितनी, आई मीन मीडिया में दूसरी चीजें भी चाहिए, बट everything would be exhausted at that time, but it will maintain the same population. Third है phage, right. So phage ये एक certain time तक grow होगी बैक्टीरिया, right, and after that the burst, right. burst होगा तो bacterial number जो है वो fall करेगा, isn't it? So the choice number second is correct. A is grown in glucose and galactose, B in glucose, and C in the infected phage, right. This is a good question actually. All right. Now, I'll take 15 more minutes, and if you understood till here, then only you can be able to understand this topic now. Lac mutational analysis, and this topic is important in the sense that in the NET examination or in the JNU entrance examination or IIT JAM examination, now they are asking questions from this portion mostly, right? That's why this portion is very important. This is very simple topic, very simple, to understand. So what happens now when the Jacob and Monod they understood the entire lab operon, right, or basics of the lab operon, then they try to understand the bit advanced you know uh, advanced mutation, bit more detail of the lab operon. So what they did actually, they made many mutational analysis, or they studied many mutational analysis of the lac operon, right? So for mutational analysis, first of all, what they made is a merozygote, right? I'm eyeing in next slide. Pe. First of all, what they form is the merozygote, right? So merozygote is nothing but the partial diploid bacterial cell they made, right? Bacteria, they are haploid actually, only one set of chromosome is there, but they made bacteria partially diploid. In the next slide we'll see. But the question is that why they are worried about the study of lac mutations. So they try to understand whether or when this operon is constitutive, when and whether this operon is uninducible, right? Whether the certain mutation is dominant or certain changes are dominant or recessive, whether the changes are cis acting element or whether the elements are trans-acting element, right? So these things are there in their mind. That's why they try to study the lac mutational analysis, right? So for the lac mutational analysis, they made partially diploid bacteria first, right? So this is the partially diploid bacteria. See here, see this is the bacterial chromosome, right? Entire bacterial chromosome on which the lac operon is there. See, wild type, okay. And here they inserted another plasmid. एक plasmid उसमें दिया, and plasmid is cloned with a lac operon only. So द लैक ओपेरॉन पॉइंट ऑफ व्यू से यदि आप देखो तो लैक ओपेरॉन की जीन जो है दो सेट में हो गई, राइट. वन सेट इज द वाइल्ड टाइप, ये वाइल्ड टाइप हो गया, और ये जो म्यूटेशनल एनालिसिस के लिए इसने रखा है दे आर देयर ऑन द प्लाज्मिड डीएनए, isn't it? So this is the merozygote. So this bacteria can be called merozygote, okay, partially diploid. All right, so see the genotype of bacteria is that. So this is the wild type, suppose. So promoter, operator, this is Z, this is Y, A, right? A नहीं दिया है तो ठीक है चलेगा, and this is the जो हमने F prime पे रखा है, means plasmid पे रखा है, right? So for this gene only it is diploid, right? So this is partially diploid. 4 cases we discuss here, 4 cases हम discuss करेंगे, और इसी में से सारे क्वेश्चंस होते हैं नेट के, एक भी क्वेश्चन इससे मिस नहीं होगा, राइट. ट्राई टू अंडरस्टैंड, जहाँ पे नहीं समझ में आए तो बोलना. सी द केस 1 हम स्टडी करते हैं केस 1 क्या है. केस 1 इज दैट द लैक operon where the mutation of the regulatory gene is there. This is very simple actually. What they did in the merozygote, this is the wild type and this is the gene which they clone onto the plasmid, right? So what they did a mutation on the regulatory gene, right? I gene पे एक mutation किया. See, they are positive means wild type. See, all the genes are here in the positive. This is wild type, okay? And here this is there on the plasmid DNA जो बाद में insert किया है prime पे, and the mutation has been made onto the I gene, right? I पे mutation किया. Mutation करने से क्या होगा यहाँ पे, what is the final or ultimate things or ultimate effect of this mutation? The ultimate effect is that no repressor would be there. I mean, mutation has been done in a way that they won't produce protein, right? Or they won't produce successful tetramer, right? They won't produce a monomer नहीं बनेगा, expression नहीं होगी, right? But they are there, but this Y type as well as this gene is there in the same cell. This I plus it synthesizes repressor, right? काफी अच्छे से repressor बनाएगा. Then they tetramerize also, ये tetramerize भी होएगा, right? And then they bind here also as well as here also. दोनों जगह पे bind करेगा. They won't differentiate that this is the Y type and this is the mutant आया है और बाहर से दिया है. No, the protein will bind at their site and the site is as it is here also as well as here also, isn't it? So they bind here. So if this tetramer is bind here and here, so what happens? It makes the entire operon switched off. So no expression would be there of this gene. I mean total operon का यहाँ भी नहीं होगा और यहाँ भी नहीं होगा, right? So no lac products are obtained, right? यदि यहाँ पे लैक्टोज नहीं हो, राइट, इन द एब्सेंस ऑफ लैक्टोज, राइट, यदि लैक्टोज यहाँ पे होता तो ये फिर यहाँ बाइंड नहीं करता है ना, बिकॉज एल्लो लैक्टोज विल बाइंड एट द टेट्रामर एंड दे इनहिबिट एंड दे प्रिवेंट द बाइंडिंग ऑफ द टेट्रामर टू द ऑपरेटर साइट. बट हियर लैक्टोज इज नॉट देयर. So in the absence of lactose, or we can say allolactose also. So in the absence of inducer, we can say here, है ना, in the absence of inducer, एब्सेंस को मैं माइनस एन से लिख रहा हूँ, इन द एब्सेंस ऑफ इंड्यूसर this operon is switched off. So what is the conclusion here? So conclusion is that both lack operons repressible, mutation is recessive. See, this is very important actually. Try to understand. Here the lack operon are repressible operon. I mean, which kind of operon? So this is repressible operon, right? Because they are repressed, and the mutation is, this mutation is recessive mutation. Why? Because this mutation has no effect on this gene. See, they are expressing their protein. They are not able to express their protein, right? Suppose ऐसा तुम समझो कि केवल यदि यही वाला होता सेल के अंदर, ये नहीं होता, तो यहाँ पे क्या होता है, ऑल थ्री प्रोटीन्स हैज बीन सिंथेसाइज्ड, इजंट इट? क्योंकि यहाँ पे रिप्रेसर बन ही नहीं रहा है. बट इसका कोई खास फर्क नहीं पड़ रहा है सेल के ऊपर इफ इट इज देयर इनसाइड द मीरोजाइगोट फॉर्म. सो दिस म्यूटेशन इज रेसेसिव म्यूटेशन. ये डोमिनेंट नहीं है. डोमिनेंट म्यूटेशन का मतलब है यदि वो म्यूटेशन हो गया, सो देन द final or ultimate effect of that mutation would be seen at the end. But here the mutation of this I gene has not been seen at the end actually, right? क्योंकि दोनों के दोनों ऑफ है ना, otherwise ये ऑन होता है ना, isn't it, this gene? So here this mutation is recessive mutation because this is fulfilled. This mutation has been complemented by this I plus gene, isn't it? This wild type I plus gene has been complemented this I minus gene. So this mutation is recessive mutation. Okay. Case 2. When the mutation has been made at the operator site. See, अब यहाँ पे देखो OC पे. See, यहाँ पे क्या किया, ऑपरेटर के ऊपर म्यूटेशन कर दिया और सब कुछ सेम है, I प्लस है यहाँ पे, है ना. So see, क्या होगा, ये भी टेट्रामर बनाएगा, इधर से भी टेट्रामर बनाएगा, दोनों टेट्रामर, ये भी टेट्रामर, ये भी टेट्रामर, राइट. बट the tetramer is not able to bind here. यहाँ पे tetramer bind नहीं हो पाएगा. Why? the mutation is there at the operator site. Okay, so this gene is transcribed and translated. यहाँ पे अपने product बनाएगा, ZYA would be there, but here no Z, no Y and no A would be there. See, अब यहाँ पे see, so this mutation is not a recessive mutation. समझ जो ये क्या है? This mutation is not the recessive mutation because this mutation is not complemented by here, right? Or it is not complemented by any other gene, isn't it? ये यदि यहाँ पे म्यूटेशन है तो देखो जीन यहाँ पे एक्सप्रेशन हो गए तीनों के तीनों. So this is called dominant. पहली चीज, first thing is that dominant. So this mutation is dominant mutation. Okay, but whether it is cis and trans, ये भी जानना चाहा जेकॉब एंड मोनॉड ने. तो ऑफ कोर्स दिस म्यूटेशन अफेक्ट्स ओनली दिस जीन, है ना, ये जो इसके जीन से इसी को अफेक्ट कर रहा है. दिस म्यूटेशन इज नॉट एबल टू अफेक्ट दिस ओपेरॉन. इसको अफेक्ट कर रहा है क्या? नहीं. That means they affect on the same stretch of DNA only. So this is cis mutation, isn't it? कैसा ये? Cis mutation है. All right. I hope you are getting. Third mutation. So third में क्या किया देखो यहाँ पे. This is very very interesting actually. They made a mutation in a way that they can synthesize the monomer. समझ जो ये क्या है, यहाँ पे IS म्यूटेशन किया है, राइट. दिस इज कॉल्ड IS म्यूटेशन. अलग म्यूटेशन, वहां था I माइनस, फर्स्ट केस वाज I माइनस, बट दिस इज IS. ओके, सो IS म्यूटेशन इज मेड इन अ वे दैट it can produce a monomer, but this monomer cannot be able to bind at the, I mean, if the if this yellow monomer can tetramerize, यदि ये yellow monomer tetramerize हो जाए, right, mutation tetramerize हो, so they don't have the site for the binding of allolactose, और inducer bind करने का यहाँ पे जगह नहीं मिलेगा इसको. I mean mutation has been done in such a way that the mutation occur in the inducer binding site. See, inducer binding site पे मतलब IPTG या फिर allolactose binding site पे mutation कर दिया, isn't it? So what happens actually? देखो यदि ऐसा करें, if it is not merozygote, only this would be there in the cell. So what happens in that case? So it has always been bind at this site, है ना, तो इसको ऑफ करके रखता है ये completely, है ना, completely this operon would be switched off, right, isn't it? Because no such binding of inducer would be there. I'm sorry, ये on, I mean inducer won't bind. So inducer won't bind, that means it is switched off. Okay, so what happens here? So this yellow one and green one both are there in the 50-50% because both are there, both are expressing, so they form hybrid, right? They form hybrid kind of thing. I mean 3 green balls would be there and 1 yellow, or 2 green 2 yellow would be there, because they are protein, and they are protein and they have the binding site for the tetramerization, right? They can tetramerize. I mean mutation has been made in a way that only the repressor cannot bind, right? जो triangle हमने बनाया था ना वहाँ पे repressor is not binding, otherwise everything is doing this protein, right? So here the repressor is not binding, right? And if the repressor is not binding, right, so it will always bind at the operator site. Even if the repressor is there inside the medium, right, even though they are not able to stop the tetramer to bind at the operator site, isn't it? So in that case what happens? Both the operon would be switched off, दोनों के दोनों switch off रहेगा, right? सी अब देखो यहाँ पे ये बहुत इंटरेस्टिंग इसलिए है कि चेंज कहाँ पे किया? चेंज इज देयर एट द प्लाज्मिड वाले डीएनए पे, राइट, बट इट्स इफेक्ट कैन बी सीन ऑन हियर आल्सो. Just a minute, I'm again sharing. Just a moment. Okay, third case we were discussing. Okay, so see what happens here. This change, I mean this IS can affect. Okay, so this IS or this mutation can affect
Sir, excuse me,
this.
sir. Sorry to interrupt you. Your screen is not visible. The
Okay,
PPT PowerPoint presentation is not visible.
not visible.
Yes, sir. It is not visible. It is showing your folder portion.
Okay, just a minute, just a minute. I
Yes, sir.
stop the okay screen. So can you see Dhwani, Dr. Dhwani?
Yes, sir. Yes, sir. Yes, sir. Yes,
Now
sir.
you can see.
Yes, sir.
Okay.
Definitely.
So see, so this is the IS. I mean, this is the mutant, and this mutant can affect this gene or expression of this operon as well as the expression of this operon, isn't it? So this is considered as trans, right? Trans, and since it is fulfilled, I mean, it is sorry, it is not fulfilled by the or it is not corrected by the any other gene present either on this operon or this operon. So this is the trans and dominant. So this mutation is trans dominant mutation. I hope you are getting. Again, I am repeating. Try to understand. See, mutation is done at the here, mutation is done here, and mutation is done in a way that they can produce a protein, right? They can produce a protein monomer and they can tetramerize also, but the inducer molecule cannot bind here, right? Inducer molecule यहाँ bind नहीं होगा. Inducer bind नहीं होगा तो क्या होगा कि this will bind here. I mean, the tetramer can bind at the operator site, isn't it? But what happens here, the concentration of this green one and the yellow one both are almost in the equal amount. Hello, yes, Dhwani. Hello. Ah, yes. Okay. So here they can, they form a combination of this green ball and yellow ball, right? Because the concentration of both green and yellow would be same, right? And if even a single yellow is there, they won't bind with the inducer molecule or IPTG or allolactose molecule. So if the allolactose or IPTG is not bound here, so what happens? They can, I mean, tetramer itself can bound at the operator site, right? And they then makes the operon totally switched off, right? So this IS can affect this operon as well as this operon, isn't it? So this is ट्रांस, राइट? ये ट्रांस हुआ, दोनों को अफेक्ट कर रहा है. ट्रांस, आई मीन सिस और ट्रांस, सिस में तो कर ही रहा है. दिस इज द सिस एज वेल एज दिस इज ट्रांस. एंड अनदर थिंग इज दैट दिस म्यूटेशन कैनॉट बी फुलफिल्ड बाय द एनी जीन. कोई भी फुलफिल नहीं कर पा रहा है इसको. सो दिस इज डोमिनेंट, राइट? सो सिस ट्रांस डोमिनेंट ये हो जाएगा. दिस म्यूटेशन इज सिस ट्रांस डोमिनेंट म्यूटेशन. Okay. The fourth case, last case, the lack of operon in E. coli with mutant regulatory gene. See, the here mutation is called minus ID, right? I minus D mutation, and this is called dominant negative mutation, right? So in this mutation, what happens actually? They mutated in a way that mutation occurs at the binding site of this tetramer, right? When this yellow ball would be there in this tetramer, so they change the binding site of this entire tetramer in a way that it won't be able to bind at the operator site. ये operator site पे bind नहीं हो पाएगा. And when it is not bound at the operator site, then both operon would be switched on, isn't it? So see, this mutation is, this is the mutation which occurs in the dominant form. I mean, this is not replaced by the any other gene, either in this operon or in this operon. So this is dominant and negative mutation, right? So because the mutant protein poisons the activity of the wild type protein. That's why it is called dominant negative mutation, right? So, see, these are the different mutations, and other mutations may also be possible, right? So, depending upon the case, you can, just manipulate your question and you can solve the question and get the answer. So, Dhani,
Thank you so much, sir. Thank you so much. Due to the constraint of time,
yeah, certainly I
uh,
can understand.
yes,
Yeah.
sir. Yes, sir. Thank you so much for providing such a great knowledge to us. We are always obliged for it, and I have got an opportunity to work with you, work under your guidance. So I know how how actually intelligent and how brilliant you are.
Okay,
So I
thank
would
you.
like to thank to you on the behalf of all the students, all the audience of the Parul University and out of the Parul University. Thank you so much,
Okay,
sir.
thank you very much, Dhani. And if
Thank
students there are any question, you collect those
Yes, sir.
questions.
I will be collecting the questions and definitely I will be communicating with you, sir.
Okay. Okay,
Thank you
thank you
so
very
much,
much.
sir.
Thank you very much for invitation. Thank you.